I'm sorry to interrupt, but before you start, there appears to only be 10 people here. Are you sure something's not up with the Zoom?
Okay,
Just want to make sure.
I don't know. My Zoom is working okay. Let me see if I can share my screen with you.
No, I trust you. It just feels like not a lot.
Yeah, I see. There are only 12 participants. Yeah, I'm surprised. Well, maybe more will join as we go. Can you see my screen now?
Yes.
Okay. All right. Let's start out with photosynthesis and respiration, because there were a lot of questions about that in one way or another. So photosynthesis produces glucose and oxygen. It consumes water and carbon dioxide. And there are two parts to photosynthesis, the light reaction and the dark reaction. So one issue was, what's the difference between photosynthesis and photorespiration? The photorespiration produces ATP. And photorespiration is a part of photosynthesis. It's in the light reaction part. So let me know if you can't see this, but there's this diagram, and all the illustrations I'm going to use today were taken from previous lectures. So you can always go back and find them in context in previous lectures. So in the light reaction, light comes in to photosystem 2, and when it does, water is split into oxygen and hydrogen. The oxygen goes off as an oxygen molecule, O2, and the hydrogen, rather than joining with a proton to make a hydrogen atom, the electron goes into photosystem 2, and it's highly energized, and so it enters an electron transfer chain. Okay, so what is an electron transfer chain? It's the passage of electrons from one carrier to another, and that's illustrated here to the left, where you have a substance, a carrier, that is reduced. It has extra electron, and it hands that electron off and becomes oxidized, goes from AH2 to A, and that electron then is transferred to another carrier, and when it does, you see at the bottom here, NAD plus goes to NADH plus an electron, and that electron is important because it can generate energy, ATP. The NADH then passes an electron back to another member of the chain, which goes from oxidized to reduced. So each of these bumps here that you see in the electron transfer chain, each of those is one of these cycles. Each one is a redox reaction. And so at each bump, energy is released. So that's why it goes downhill, because the electrons are becoming less energetic as they go because they're losing energy. And then we go to photosystem 1, and again, a photon of light comes in, re-energizes electrons, and they go into another electron transport chain. So again, you have the recycling, and out of that comes NADPH, which is then used later. So what's happening, and that all of this is going on in the chloroplast. in the thylakoid membrane, which has within it the thylakoid compartment. So hydrogen ions, while all this is going on, hydrogen ions are accumulating in the inner thylakoid compartment, which makes a gradient between inside the compartment and outside. A lot of you asked about chemiosmosis. Well, chemiosmosis happens within the membrane, and it's coupled to the electron transport chain. And so this little diagram here at the top right with the blue dots, it illustrates what chemiosmosis is. The definition is off to the left. It's the movement of ions across a semi-permeable membrane down their electrochemical gradient. And when this transfer, this movement occurs from high concentration in the inside of the membrane to outside, the enzyme ATP synthase gets involved and converts a molecule of ADP to a molecule of ATP. And that ATP then goes to the Calvin cycle, which is the other part of photosynthesis. It's the dark reaction, and that's what fixes carbon and makes glucose. So is it clear? There were a lot of questions either directly about this process or related to it. Any questions about this light reaction and how electron transport is coupled to chemiosmosis, and it's really the chemiosmosis that generates ATP in photophosphorylation. And remember, we're going to talk about oxidative phosphorylation, which occurs then in not in the chloroplast, but in the mitochondrion. Okay. So to take the information from that figure and make a summary of it, what you see here, we have both the light reaction on the left and the light independent reaction, or the dark reaction, on the right. The light reaction occurs in the thylakoids. In the chloroplast, the dark reaction occurs in the stroma of the chloroplast. So this whole object here is the chloroplast. So the summary is that water and carbon dioxide go into the chloroplast, oxygen and sugar come out. So the ATP that is made in the light reaction is used in the dark reaction to generate sugar, to lock energy into chemical bonds. All right, the dark reaction, so that's the Calvin cycle here on the right, is elaborated in more detail here. And you don't have to know all the steps and all the molecules in the Calvin cycle, but what you do need to know is that the ATP that's generated here, this ATP goes into the Calvin cycle and is used in this step here, 6 ATP go to 6 ADP, and the energy from that is used to fix carbon. And there's 3 more ATPs used over here. So each time the Calvin cycle turns, it uses 9 molecules of ATP, and out comes the bottom here, and I've circled it, glucose. And glucose is food. It goes to the Krebs cycle for oxidation and the release of energy to do work within the cell, and that is respiration. So that's the
Professor?
Kelvin cycle. The Kelvin cycle is the dark reaction of photosynthesis. And I put in another illustration of chemiosmosis. Doesn't really add anything except it is bigger and bolder. So in the middle you see a membrane, and you see transporters of hydrogen ions from one side of the membrane to the other. So the concentration at the top is higher than the concentration on the other side of the membrane. And then chemiosmosis is circled here. It involves a channel and the enzyme ATP synthase, which takes a molecule of ADP plus inorganic phosphate and makes a molecule of ATP, which has a high energy bond that can then be used to generate energy to run, for example, enzyme reactions. There were a lot of questions about chemiosmosis. Is everybody clear now what it is and how it fits into the big picture? These are answers to two particular questions. Chemiosmosis is not involved in substrate level phosphorylation, but it is required for both oxidative phosphorylation in mitochondria and photophosphorylation in the chloroplast. Okay. The other side of the coin from photosynthesis is respiration, which produces carbon dioxide and water. It consumes glucose and oxygen. Here is an overview of what happens in respiration. Starts with glycolysis. So actually starts with glucose, which undergoes glycolysis. And what glycolysis does is to break glucose, which is a 6-carbon sugar, into 2 3-carbon molecules, which are pyruvate. So glycolysis requires 2 ATP molecules in order to proceed, but because there are 2 points of substrate level phosphorylation during glycolysis, there's a net yield, a total yield of 4 ATPs, a net yield of 2 ATPs from glycolysis of 1 molecule of glucose. So the glycolysis also generates electron carriers along with 2 molecules of pyruvate. These both cross, and this all happens in the cytoplasm. Glycolysis happens in the cytoplasm, not within an organelle. These molecules then cross both the outer mitochondrial membrane and the inner mitochondrial membrane. It's the inner membrane where most of the action occurs. The electron carriers go into the electron transport chain within the inner mitochondrial membrane. And that, just as in photosynthesis, that electron transfer process is coupled to chemiosmosis. So that again, you have ATP synthase generating ATP, generates a lot of it because a lot of electron carriers are generated from glycolysis. Now the acetyl, the pyruvates are converted to a molecule called acetyl-CoA, and that's the first step in the Krebs cycle. The Krebs cycle has 2 substrate level phosphorylations to generate 2 molecules of ATP. It releases carbon dioxide. Respiration generates carbon dioxide. It takes up oxygen, which you see here, And the electron transport chain coupled to chemiosmosis generates 32 ATP molecules from one molecule of glucose at the start. And so the total number of ATP molecules would be 32 plus 2 from the Krebs cycle, a net of 2 from glycolysis, for a total of 36. Some calculations show 38 molecules of ATP from one molecule of glucose. And what is done with all this ATP? This is what drives all the chemical, the enzyme, all the enzyme reactions that go on in the cell. Thousands and thousands of reactions. So this is the counterpart of photosynthesis. This is respiration. And I hope it's clear how the electron transport chain coupled with chemiosmosis is involved in both processes. This picture just summarizes or illustrates an overview of the process. We have glycolysis. One molecule of glucose makes 2 molecules of pyruvate in the cytoplasm. Pyruvate crosses the mitochondrial membrane. It's converted to acetyl-CoA, which is the first step in the Krebs cycle. The Krebs cycle then generates a little ATP, but what's more important is the electron carriers that it generates and the electron transport chain and the inner mitochondrial membrane coupled with, and this little diagram here always indicates chemiosmosis. Those two together generate 32 to 34 molecules of ATP. Then I included this just to compare oxidative phosphorylation, which is respiration, with photophosphorylation, which occurs in photosynthesis. So in oxidative phosphorylation, it produces ATP using enzymes and oxygen. It's the last stage of aerobic respiration. Photophosphorylation produces ATP also, but the source of energy now is not glucose, it's sunlight. The energy source is oxygen and glucose in the Krebs cycle, sunlight in photosynthesis. Oxidative phosphorylation occurs in the mitochondria, photophosphorylation in the chloroplast. Oxidative is in cellular respiration, photophosphorylation is in photosynthesis, and the final acceptors are different in the two. So that's included just to help you think about it. Here's another summary comparing the Krebs cycle and the Calvin cycle. Krebs cycle is a part of respiration. Calvin cycle is in photosynthesis. In two different organelles, Krebs cycle in mitochondria, Calvin cycle in chloroplasts. One is catabolic. Krebs cycle takes glucose apart and releases energy in doing so. The other is anabolic. It puts things together and builds glucose. In Krebs cycle, ATP and carbon dioxide are produced. In another case, they're used. Respiration occurs in all aerobic organisms, whereas the Calvin cycle occurs only in plants because it occurs in chloroplasts and only plants have chloroplasts. Krebs cycle oxidizes glucose, Calvin cycle creates it. And in the case of the Krebs cycle, it's aerobic, whereas oxygen isn't involved in the Calvin cycle. Okay, there was a question what would life be like without photosynthesis? Interesting question. We don't know because we have photosynthesis, but we have one example of life without photosynthesis, and that's chemosynthesis, which occurs in these deep ocean vents. And the life forms are very primitive that live by chemosynthesis. So I would imagine if photosynthesis had never evolved on this planet, life might have evolved anyway at these deep ocean vents, which would have occurred. And that might be the end of life on this planet. It's alive, but it's not doing much. It certainly wouldn't have evolved in the same way that life has evolved with photosynthesis, because photosynthesis is so efficient at capturing energy and capturing light energy and making chemical energy out of it. There was a question about stomates. So what do stomates do? They regulate the exchange of gases in leaves. And the main gases are oxygen, carbon dioxide, and water vapor. Where in a plant does photosynthesis occur? We know it occurs in the chloroplast, but where are the chloroplasts? Any green part of the plant can photosynthesize. That's mostly in leaves. And if you take a cross-section of a leaf, which you see here, there's an upper surface and a lower surface. And in between are what are called mesophyll cells. And within a mesophyll cell, you find chloroplasts. Most of the stomates, not all, but most of the stomates are on the lower surface of the leaf. They're little openings that are oval shaped. There's a cell on either side of the opening, and that cell can swell or shrink. If it swells, it closes the opening. If it shrinks, it opens the opening. And so the stomates open and close depending on the conditions. So if conditions are dry, the stomates will close to prevent the plant from losing moisture. If it's wet, they do the opposite. They open, let moisture out. And of course, they need to let out carbon dioxide as respiration occurs. And respiration occurs both, and this is important, respiration occurs in the chloroplast as well as in the mitochondria. And there was a question about ATP, ADP interconversion. So that's why I put this illustration here. The structure of ADP is at the bottom. ATP is at the top. So what happens when ATP synthesis is involved, ADP, which has two phosphate groups at the 5' position, takes one inorganic phosphate group and adds it on so that ATP, triphosphate, is what you see at the top. And the bond between the third and the second phosphate groups is a very high energy bond. So when this bond is broken, energy is released. And that's why ATP is the energy currency of the cell. If a reaction needs energy, ATP allows one of its most external phosphate bond to be broken to release the energy that is needed for the reaction. So the conversion from ADP to ATP is endergonic. It requires energy. When ATP is hydrolyzed, energy comes out. It's an exergonic reaction.
You say when it's hydrolyzed?
No, I shouldn't have used the word hydrolyzed. When the bond is broken. Okay, Scott, are you still there?
Yes, I am.
Would this be a good time for you to do inheritance, or
Sure.
would you prefer later?
No, I could do it now.
Okay, then I'm going to stop sharing my screen and you can start sharing yours.
All right. So the questions were kind of along the lines of what was a genotype, what was a phenotype? That was one of the things. And you do have to have these terms down because they're important. A phenotype is a descriptor, a descriptive word or phrase for a particular trait, something you can see and then describe to someone else, like brown or long or tall, something along those lines. But it's a descriptive word. That's your phenotype. Genotype is talking solely about the alleles and their combinations that you would get when you do Mendelian inheritance. You're going to use just letters for that. That's going to be your big T, little t, or big H, or small c, small c. So genotype, it's really telling you what are the types of genes, alleles, that this particular individual has. Remember, there's always 2 alleles for every trait in most organisms. Certainly in the eukaryotes that we're typically thinking about, you're going to have 2 alleles for each trait, one from a mother, one from a father, or the male and the female. But so remember, genotype, you're just dealing with the letter assignments, the big T, little t type thing. Phenotype, you're using a descriptive word. Another question was along the lines of F1s, F2s, what are they? Well, that's important because when you're working out Punnett squares, sometimes they will ask you in the problem, what is the genotypic ratio of the F1 generation? What is it for the F2 generation? So those are important as well. You start out with 2 parents, a male and a female. When they are crossed, and cross is just another word for bred or mated. So when these things are crossed, the first generation of offspring produced are known as F1s. And any subsequent mating of F1s will then be known as F2s. And matings between F2s would then become F3s, their offspring. So it just follows. So you start with a parent, and the first set of offspring that come out, F1. F is for filial, which refers to like son. So they're just talking about offspring. And the number is just which generation you're speaking of, the first, the second, the third, etc. So when you're doing your dihybrids or your monohybrid Punnett squares, really what you're coming out with is you're mating 2 parents and you're going to come out with an F1 generation. Then you may have to take these F1s and do something with them. Maybe you'll cross them with one another and you'll produce an F2. There's also the self-cross, which is mating a single individual with itself. Test cross, where you're, or back cross, where you're going to determine heterozygosity or homozygosity of an individual based on mating back with a recessive parent. All right, so you get the Punnett square problems, and I have, let's see, I cannot share screen.
You can't?
I cannot.
Okay. Well, would you like me to share mine and you can use the monohybrid and dihybrid crosses that I have?
Sure. Yeah, that would probably work. That'd,
Okay. All right, then I'll.
and somebody's already asked if you could send that document to them, so they would be able to see it and
Yeah.
then review
I
it.
noticed the request was for a PDF. Does that mean that the Word document's not working?
On the first few pages, the images kind of turned out weird. Like it's not where it is on your screen.
Okay. Yeah, Canvas often does that. Okay, I will, after the session is over, I will send it. I'll convert it to a PDF and send it that way. All right, can you all see the monohybrid cross?
Yes.
Okay, so Scott, hopefully you can see it and describe it.
All right. So the two terms you're pretty much going to hear about are monohybrid crosses and dihybrid cross. And what is the difference between the two? With a monohybrid cross, you are simply looking at one trait, be it fur color or stem length or wing size or something along those lines. You're simply looking at one trait at a time. Dihybrid, you're looking, di means two, so dihybrid, you're looking at two different traits where you might be looking at wing shape and eye color at the same time, or flower color and seed shape at the same time. So monohybrid, one thing. Mono means one trait. Dihybrid, we're looking at two traits at the same time. So the first problem you see here is a monohybrid cross. So we know we're going to be looking at one trait at a time here. And this is talking about fruit color in apples. And they're kind enough here to tell us that it happens at a single genetic locus, which is good because it tells us that we're not dealing with anything overly complicated, like a polygenetic inheritance. So it tells us that green fruit color is dominant to the pink fruit color. And remember, we can assign any arbitrary letter that we want to this trait that we're looking at. In this case, they've chosen big G to be green and little g to be pink. We always use a capital letter for the dominant trait, and we always use a lowercase letter for the recessive trait. Also, you must use the same letter for each trait. You can't do like a big G and then a small p for pink. That will not work. You have to use big G, little g, or big P, little p. It has to be the same letter for the same trait. So we have arbitrarily assigned capital G for green, lowercase g for pink. If an apple tree is homozygous for green, which means it's homozygous, it expresses the same allele, the same exact allele on that locus. In this case, for green, we know that that would be big G, big G. Homozygous, it has the same allele variant at that locus. And then it's crossed with a homozygous for pink, which again, we know that homozygous means we have the same allele variant at the same locus. So that would be small p, small p. Remember, we have to have
Small g, small g.
small g. Pardon me, I screwed myself up there. So small g. Remember, we have to have two copies of an allele, or two alleles at each locus for all the individuals that you're going to worry about in this class. They want to know the genotypes and the phenotypes of the F1 and the F2. So here's why we need to know what all those terms mean. So simply, they're asking us, what are the allele combinations and what are the descriptive words for the traits of the first generation offspring, and then when we cross two F1s, the second generation offspring. So here's our setup. You can see big G is green, little g is pink. Parental cross, we knew that one parent was big G because it was homozygous green. The other parent is small g because it was homozygous pink. In the F1s, you will get all heterozygotes. And if you lay out a Punnett square, similar to the one you see here, but if you put big G on the left side, big G at the top, big G at the bottom, and across the top, if you had little g on the left and little g on the right, when you did your Punnett square, you would find every single F1 will be a heterozygote of big G, little g. Remember, heterozygote means you have one of each of the alleles. So, for that trait. And if you ever see homozygous breeding with homozygous, homozygous dominant with homozygous recessive, as a tip, they will always be the F1s will always be 100% heterozygotes. You don't even have to work out a Punnett square. It's just the way it will come out. You can work one out if you don't believe it, but all your F1s will be heterozygote, big G, little g. So then they wanted, that's our genotype for the F1 is big G, little g. They want to know the phenotype, that's our descriptive word. We know that green is dominant to pink. As long as they have one dominant allele, that is sufficient to express the dominant trait, because they don't mention any other type of inheritance. And you always default to complete dominance unless it's given to you somewhere else in the problem in some fashion. This one does not. So a big G is sufficient to express the green color. Therefore, our phenotype will be all green fruit. Now, that's for the F1s. We have to do the same thing for the F2s. F2 is simply if you take two F1s and cross them together. We know that each of the F1s were heterozygotes. So now this Punnett square that you see actually applies, where we have on the top a big G and a little g. That's one heterozygote F1. And on the left side, we have a big G, little g. That's another heterozygote F1. When we do our crosses, we will get from in the top row here, if we look at the first column, big G, second column will be big G and little g. When we look at the second row, we'll have a little g, and the first column gives us big G, little g, and the second column gives us little g, little g. This is what we would expect to find when you are crossing two heterozygotes. You will have one homozygous dominant, two heterozygotes, and one homozygous recessive. You'll always have that. That's the 1 to 2 to 1 ratio that they talk about. So now they want to know what we know our genotypes. We've already figured those out. What are the phenotypes? Well, big G, two dominants, it's going to express green. A big G, little g, because the problem doesn't state otherwise, a single dominant allele is sufficient to express the dominant trait. So we will have big G, little g will be green, just the same as it was up above. A homozygous recessive, small g, will result in the pink fruit color. So this would be, you would have big G, one of them. You'd have big G, little g, two of them. Little g, one of them. That would be your genotypes. Phenotypes, we would have 3 green and 1 pink. Remember, phenotype is the descriptive word. Is there any questions? I know this is like going quick, but you've seen these before, but are there any questions? Do you not understand any particular thing we talked about here?
How do we get both the F1 and an F2 generation?
You have to do them in steps. First, you have to cross 2 parents, and that will tell you what your F1 generation is. Then you will do another Punnett square, and you will lay out two, and that's actually this particular one that you see is actually the second step where you've crossed two different F1s, and this gives you your F2. But you would have to lay out a Punnett square for each generation, ideally. Did that answer your question?
Yes.
Okay. All right. Nobody else has questions or yes? Okay. Well, let's roll down to the dihybrid. Are we going to be tested when the colors mix? I think it's called codominant. That comes from Rachel.
Scott, look at the chat. I think so. Somebody have a question too.
Yes, I was just reading that. Thank you, Amir. Rachel, it is fair game. That's the best I can tell you. So there are two different other types of dominance. One is the codominant, which you talk about, which is a mixing or a blending of the two colors. For example, red. If red was one color and white was another color, the mix in codominant would come out pink. Or I'm sorry, that's incomplete dominance. Codominance would be when you have a red and a white, and your offspring have red and white in them, not pink, because they don't mix. They are both expressed. They're co-dominant. They share dominance. So both of them, both of the colors would be shown up on one individual offspring. So in that last problem, if we were looking at the F1s where we had 100% heterozygotes, they would be red and green, or red and pink, green and pink at the same time on each individual. There would be no actual blending. Did that make sense? So incomplete dominance, neither one of them wins, so they come out in the middle somewhere. Codominance, they both are dominant. They're sharing dominance, so both colors appear at the same time in the same individual. Okay. I'll keep an eye on chat, Rachel, if you have anything else you want to type in there about that explanation. So a dihybrid cross, we're talking about two different traits at the same time. We have to consider them both. These are going to be your potentially big Punnett squares. They can have up to 16 boxes depending on how evil the professor's problem setup is. In this case, he gave us the full Monty. We have a true breeding brown horse with a fuzzy mane crossed with a white horse with a straight mane. Assume that brown color is dominant to white and fuzzy manes are dominant to straight. So you have to read between the lines a little bit here. This is a true breeding brown horse. It means it only breeds, ever only breeds brown and fuzzy. And this other horse, it doesn't really tell us what's going on there, but we do know that if it's straight mane, and straight manes are not dominant, they are recessive. So if this has a straight mane, that must be a homozygous recessive for mane, hair straightness or fuzziness. And if it's white, then it has to have it has to be homozygous recessive for color. So this horse, we know this would be, well, we'll get to the letters down here, but we know this horse is homozygous recessive for color, homozygous recessive for hair, mane hair thing. But the true breeding horse, this is another way of saying homozygous dominant, or true breeding really is telling you it's homozygous. In this case, brown is dominant color, so it's homozygous dominant. And we would assume it's also homozygous dominant for the mane as well. All right, so they want to know genotype and phenotype of F1, genotype, phenotype of F2. So again, we're setting up our basic Punnett square starting out. We have to assign our letters. We know that brown is dominant, so we'll assign that a capital B. We know that white is recessive, so we'll assign that to a lowercase b. We know that fuzzy mains are dominant, so we'll assign that to a capital F. And we know that straight mains are recessive, so we will assign that as a lowercase f. You do your parental cross. If you do the FOIL method, or however you want to come out with the gametes, you will find that the true breeding brown horse with a fuzzy mane, capital B, capital F, can only produce a capital B, capital F gamete. There are no lowercase letters in that particular parent, so they can't produce any gametes that are not a capital B or a capital F. Similarly with the the white horse, since white is recessive, it must be homozygous recessive for color and for the mane. Therefore, we give it a small b, small f, small f. And if you again do FOIL or however you work out your gametes, you'll find that it can only produce a little b, little f. Similarly to the last problem, we have all dominant and all recessive, so our F1s are going to be heterozygotes. One parent is completely dominant, one parent is completely recessive. Therefore, we're going to have heterozygotes in the F1 generation. That's all we will get. If we did our Punnett square, we would come out with a big B, big F, little f. The phenotype of the F1, because it has one capital B, they don't tell us anything different about the pattern of inheritance. We're going to assume complete dominance. One dominant allele is sufficient to express that trait, and that means that we'll have a brown horse. Same with the mane. We have one dominant allele that is sufficient to express the fuzzy mane. So our F1 phenotype, the descriptive term, is all brown with fuzzy manes. Now it gets a little more complicated because we're going to breed two of these F1s to make our F2 generations. So we have to figure out what particular gametes these horses can provide. And if you, again, you do the FOIL method or however you lay out your gamete combinations, you'll find that we will get one big F, big B, big F, one big B, little f, one little b, big F, and one little b, little f. So since each parent is the same, we lay out the Punnett square with one parent across the top, one parent's gametes down the side, and then we simply do our normal Punnett square thing where a big B results in a big B down here, big B in the first F1 box, top left, a big F, we're going to have two capital Fs here. So this first offspring will be big B, big F. You just continue matching up your letters through all 16 boxes here. And then you have to start to count things up because they want to know about the genotypes. And so they're going to want to know quantities, just like in the last one. And for phenotypes, they're going to want to know quantities. When you look at your F1 genotypes, if you tabulate them all up, you will find these numbers here. I won't go read through them all. But for the F2s, they typically will show you when you're talking about breeding two heterozygote for both trait individuals, a 9:3 to 3:1 ratio of brown fuzzy. If we counted them all up, we would have 9. Brown with straight, we would have 3. White and fuzzy, we would have 3. White and straight, we would have 1. You're going to have only one completely recessive individual, homozygous recessive for both traits.
Yeah, good, Scott. Does anyone have any questions for Scott about a dihybrid cross?
Don't get thrown when you see this dihybrid. It looks intimidating. Thank you, James. Looks intimidating, and it looks like it's a ton of work, but it really isn't. It's just matching up letters. So don't get freaked out when you see a dihybrid cross. It's really not asking you to do anything you don't know how to do. You just have to, push the pencil around on the paper a little bit more is all.
Okay, thanks, Scott. That was good. If there are no more questions about Mendelian inheritance, I'd like to go back to where we were.
I'm going to be stepping away for a few minutes, sir, but I'll be back.
Okay. Let's see. Okay. There were a lot of questions about enzymes. One question was about whether all biological reactions depend on enzymes. The answer is yes. And why is that? Because enzymatically catalyzed reactions occur very quickly in fractions of seconds. Whereas if there was no enzyme there, most reactions would take hours or days without enzymes. And that's way too long for living biological systems. Other questions about cofactors. Most enzymes require one or more cofactors. And these are either metals or organic compounds like vitamins. The question about enzyme and substrate shapes. So that you see here with the blue and the green. The blue is an enzyme, the enzyme sucrase, which converts sucrose, the disaccharide, to two monosaccharides, glucose and fructose. So sucrose has to fit into the shape of the enzyme. If it doesn't, the reaction doesn't work. So important point is shape is critical. I didn't put it in here, but the lock and key analogy is appropriate when you think about shape. There were questions about enzyme inhibition. And these are very important questions because it's enzyme inhibition that regulates what's going on in the cell. It's whatever the organism needs, enzymes are inhibited either a lot or a little or not at all, depending on what is called for elsewhere in the cell. So there's reversible and non-reversible inhibition. And types of reversible can be either competitive or non-competitive. You see that illustrated here, competitive inhibition. You have a competitive inhibitor, it has the same shape as the substrate. And so it will fit into the active site of the enzyme. And if it does, it occupies all the space the substrate can't fit in. So if the concentration of substrate is very high and the inhibitor is low, then most of the time the substrate will fit in and the reaction will occur. If the reverse is true, then the reaction slows down because most of the enzyme sites are occupied by the inhibitor. Now, non-competitive inhibition, the inhibitor in green and the substrate, they're not competing for the same site. The inhibitor has its own site at a different part of the molecule. And if it occupies that site, it changes the shape of the active site of the enzyme, so that now when the substrate comes along, it has nowhere to fit. So as long as that inhibitor is attached to the enzyme is the wrong shape, and the substrate can't fit, there is no reaction. So if in a mixture, some of the enzymes, say half the enzymes were inhibited, and half were not inhibited, then the reaction rate would be half speed because only half of the enzymes are working. So is that clear? There were several questions about inhibition. And a very important type of inhibition is end product inhibition. That's shown here. If you start on the left, the blue box is a substrate. And the first enzyme in the pathway, the first step, converts the pale blue box to a dark blue box, and on and on. Each enzyme in the pathway converts the box to something else. And finally, you end up over on the right with a red box, which is the end product. So this end product is used by the cell somewhere else, and the demand for it depends on the needs of the cell. So if there's very little demand, the concentration of this red box, this end product, will increase. It will get to be very high, and some of this end product will go over here, and it has a site, an attachment site, in enzyme number 1, the first step in the pathway. So if the end product binds to enzyme number 1, that reaction doesn't occur. And if that reaction doesn't occur, none of them do. Because pathways go in steps, and they have to go in the order prescribed. So if the concentration of the end product of the pathway is high, that end product will inhibit the first step and slow the pathway down, so that now end product is being produced at a much slower rate. On the other hand, supposing the cell has a great need for this end product right now and uses it all up, well, now there is nothing to block step 1, so this enzyme, this pathway will run at full speed and will make a lot of end product, which is being gobbled up by the needs of the cell. So is that okay? Anybody have a question about how feedback inhibition works? Well, there are a couple of questions about how you read a graph. So I just chose a picture of 2 graphs to describe how graphs work. A 2-dimensional graph, which both of these are, has 2 axes, an x-axis, which is horizontal, and a y-axis, which is vertical. And those meet at a point, and that point is usually indicated as 0. So on the y-axis, in this case, we're talking about enzyme reaction rates. So if we actually quantified the reaction rates, you'd have numbers here, a rate of 100, rate of 200, and so on, rates increasing as you go up the y-axis. On this top graph, on the x-axis, we have temperature increasing from 0 to 100 degrees. And so if you plot the rate of reaction at any given temperature, say if the temperature is 20, then you go up to the line, and that indicates the rate at a temperature of 20 degrees. So as temperature increases, the reaction rate increases up to a point, and this point is called the optimum rate. So approximately 38 degrees is the temperature at which this enzyme runs as fast as it can. If you go any lower than that, it slows down. If you go any higher than that, it slows down. So optimum is the fastest rate that it can go. And you see the same thing with the purple line. except it's a different enzyme. And so the optimum is at about 75 degrees. Lower than that, the enzyme is running at a slower speed. Higher than that, it denatures. And so if you go to the graph at the bottom, it's the same thing. The rate of reaction is on the y-axis. In this case, the x-axis is showing pH. And pHs can run from 0 to 10. And depending on the enzyme, in the case of pepsin, the pH optimum is 2. So if the pH is lower than 2, the enzyme runs slower. If it's higher than 2, denatures and the rate goes down. Go to a different enzyme, trypsin, it just shifts to a different place on the scale. So the optimum for trypsin is about 8. If the pH is lower than that, the enzyme runs slower. If it's higher than that, the enzyme runs slower. So is that clear about how you read a graph? Okay. Enzyme denaturation. Several questions about this. And the definition of denaturation is indicated here. Loss of quaternary structure, tertiary structure, and secondary structure causes denaturation. And it's caused by stress, such as a strong acid or base, a salt, an organic solvent, radiation, or heat. Any of these things can cause enzyme denaturation. Now, how does denaturation happen? To think about that, you have to go back and think about the enzyme, the protein structure, enzyme structure. So you have primary structure, which is the amino acid sequence. Secondary structure is composed of beta-pleated sheets and alpha helices, and these are held together by hydrogen bonds. Tertiary structure is where the chain of amino acids gets all twisted up into a particular shape. And quaternary structure is where more than one protein chain is involved. So if you look to the right here, tertiary structure bonds, there are 4 different types of bond that can hold the tertiary shape. Hydrogen bonds, hydrophobic interactions, ionic bonds, and disulfide bridges. So if you abuse this thing with heat or radiation or strong acid or strong base, what happens? You break these bonds. You remember, all of these bonds are weak except for disulfide bridge, which is strong. It's a covalent bond. But these other bonds are fragile. They're easily broken. And so if the environment is not conducive to these bonds holding, they will break and the tertiary structure is destroyed. And if the tertiary structure, remember, the shape of the molecule is critical to its activity. If the tertiary structure is damaged, the shape of the enzyme changes and it can no longer fit its substrate. So that's enzyme denaturation, that's what it is. And these 2 figures show how it works. No questions? All right, there were a couple of questions about leaf color. Most of the time you look at a plant, it's green. Look at a tree, it's green. There are 3 reasons why it might not be green. I listed them here. The chlorophyll, it could be green, but the chlorophyll is masked by another pigment, and so it might look red, like a red maple. If the plant is not green, it might be parasitic. It might actually have no chlorophyll, and it's synthesizing other pigments, so it has a color. It might be orange, it might be yellow, but it's never growing by itself. A parasitic plant is always sitting on a green plant, deriving its food sugar in particular, from the green plant. Because without chlorophyll, a parasitic plant cannot make its own food. Or you may see a non-green plant because of environmental change. So for example, in the fall, which is where we are right now, there's lower light and lower temperature, and that causes chlorophyll to break down. And other pigments can be synthesized instead. And that's why you see a loss of green, because it's actually disappearing, and other colors are being synthesized. These could be reds, carotenoids, could be yellow or orange. There are also browns. So those are the three reasons why a plant that you look at might not be green. All right, the coronavirus question that I asked, and a number of you had issues with this. It wasn't so much about coronavirus, but rather how you think about a problem. Now, I had not, at the time I asked this question, I had not talked about a vaccine for coronavirus. And yet I asked you about a vaccine, and I gave you the information that we had at the time. The pharmaceutical company Pfizer had made an announcement that they had a vaccine and that it was not a protein. So that's the information I gave you. And at first glance, you would say, I have no idea. But then what I was hoping to get across is that you don't just throw up your hands and quit. You say, is there anything I know that might be relevant to this question? And going back to what we went over a couple of times in lecture is the structure of a coronavirus particle. We know that it has these spike proteins on the surface, and we know that its genome is made of RNA. Those are the two things that were emphasized. And so with respect to the question, we know it's not spike protein, it's not the vaccine because it's a protein. And we know that the vaccine is not a protein. So what's left? RNA, the genome. And so that was what I was looking for, is that thought process, that deduction to arrive at an answer. Now, you could argue that there's more to this virus than just RNA and protein, because, for example, it has membrane. And if you wanted to make a case that it might be a component of the membrane, say a phospholipid, that is antigenic, and the vaccine is, and that is the vaccine, I accept that argument. Nobody did make that claim, and so I didn't have to deal with it. But if you make a logical argument for an alternative, that's acceptable. That reflects good thinking. Okay, somebody asked a very interesting question. We know that a number of syndromes are linked to the X chromosome, the sex chromosome. But what about Y? Well, it's interesting when you think about it, because the Y chromosome only exists in males. It's very small. It only has about 200 genes. And most of these genes, if you delete any one or a group of them, they cause male infertility. And that occurs about once in every 2,000 men. They're male infertile. Therefore, it's not heritable. So when a mutation occurs in Y, that's the end of the road for that chromosome. It's not going to be inherited because it causes infertility. So there are very few Y-linked traits for that reason. So thank you, whoever asked that question, because it raises an interesting point. Evolution. Someone said, what if bacteria become resistant to all antibiotics? It's a very good question because it's happening. It's a huge problem, for example, in hospitals. Nosocomial infections are the infections that you get while you're in the hospital, and they're caused by antibiotic-resistant bacteria. And why is this such an enormous problem? It's because antibiotics are overused. They're used way too widely. For example, animal feed. If you put an antibiotic in food for cows, they make more milk. So farmers put antibiotics in cow food. Sometimes some computer keyboards have antibiotics incorporated into their keys so that when you're typing, you're supposedly free of bacteria on your fingers. Hardware such as doorknobs, disinfectants like hand creams. So antibiotics are everywhere, and what that means is it gives bacteria lots of chances to mutate and become resistant to them. So the point to be made about evolution of resistance is antibiotics should only be used when they're needed. And usually that means to fight an infection that's already occurred. You don't use them preemptively to try to make a sterile environment for ourselves, because that's why we have immune systems. Our immune systems have evolved to protect us against bacteria and viruses. We don't need to spread antibiotics all over. Another question was, why is adaptation reversible? This is adaptation as opposed to evolution. Well, adaptation is a short-term change in response to an environmental change. And one of the examples I gave was animal coat color. Remember the Arctic foxes that are white in the winter and brown in the summer? That is a temporary change. Animal changes color for a season, and then when the next season comes, it changes back again. So it's reversible. And the point here is that the capacity for seasonal adaptation is indeed inheritable. In other words, these foxes inherit from generation to generation the ability to change color. So that's heritable. But the season adaptation itself is not heritable. In other words, when you change from brown to white, the next generation isn't all white. Next generation still has the capacity to switch back and forth. So that's the distinction between an adaptation and evolution, which is permanent. When an evolutionary change occurs, it's heritable generation after generation after generation. There were a couple of questions about Mendel and Darwin. What's their connection? Well, Darwin contributed the concept of evolution and was based on natural selection, survival of the fittest, and that's how new species originate. What did Mendel do? He provided the rules, a mechanism for what Darwin was proposing. And the mechanism is that genes are discrete entities that control phenotypes. And I'm running out of time here, but just to make the point clear, at the bottom of this, I have red times white. Prior to Mendel, progeny were thought to be a blend of the parental phenotypes. So if you had a red one and a white one, and you crossed them, you'd get pink. And that actually happened in most of the crosses because they weren't starting with homozygous lines. And that was part of Mendel's genius that he did, so that when he crossed red times white, he didn't get pink, he got either red or white. So Mendel showed that progeny possess sets of parental traits, not a blend of parental traits. Okay, the time for this class is over, so you can leave if you need to. I will send a PDF of this document later, but any of you who want to stay, I still have a few more questions to answer, and I'll do that as long as anyone hangs around. Remember that Scott's workshop starts at 2. Someone asked, what's the difference between a prokaryote and a eukaryote? Well, there are quite a few differences, and some of them are listed here, certainly not all. But prokaryotes don't have a nucleus. Prokaryotes do not have organelles of any type. So what do they have instead of a nucleus? They have what's called a nucleoid, which is simply a single very long loop of DNA floating in the cytoplasm, whereas eukaryotes have a nucleus with chromosomes. Prokaryotes, how do they propagate? Their cell division is called binary fission, and that results in identical daughter cells, no genetic variation. Whereas in eukaryotes, they have mitosis, which gives identical daughter cells, but they also have meiosis, which is designed to introduce genetic variation. And we've talked a lot about that. Okay, eukaryotes have endoplasmic reticulum, which has the ribosomes. How do prokaryotes generate their energy if they don't have organelles? Well, they do it across their plasma membrane, whereas eukaryotes use the mitochondrial membrane. Most prokaryotes are single-celled. Most eukaryotes, not most but not all, are multicellular. The prokaryotes are almost entirely bacteria and archaea. Eukaryotes are plants, animals, and fungi. And finally, prokaryotes always have a cell wall. In eukaryotes, it's optional. Plants and fungi usually do. Animals do not. And I put in a picture here illustrating what's on that list. Somebody asked a question about how does tRNA work? And so I've put in a picture here of a tRNA molecule. How does it work? On one end of the molecule, this green end down here is what's called the anticodon. And on the other end of the molecule, the red part is what's called the receptor end. That's where an amino acid is attached to this tRNA molecule. Now, mRNA is a string of bases. And they arrange themselves into sets of 3, and these are called codons. So the codon and the anticodon match up, and that matchup is critical in determining the amino acid that's going to be added to the growing chain. Remember that the ribosome is going along the mRNA, calling for tRNAs to add the next amino acid. So in this case, the mRNA codon is GCC. So the anticodon on tRNA would be the complement, CGG. And this happens to be the codon for alanine. So the amino acid up here on this end is alanine. Therefore, when the anticodon binds to the codon, alanine is incorporated into the growing amino acid chain. So is that clear how tRNA specifies a particular amino acid? Someone asked, does CRISPR cause cancer? You can't prove a negative. You can't ever prove that CRISPR doesn't or could never cause cancer, but the chances of it are extremely unlikely. And let me just remind you what CRISPR does. It causes any mutation in any organism, from bacteria to whales. And it's site-specific and it's universal. And it's this site specificity that is one of the remarkable things about it. And how does that happen? Well, to remind you, to start CRISPR, you make a guide RNA. You synthesize this in the lab, and this is like a 15 base pair RNA, which is specific to the site in the genome that you want to mutate. So it won't go anywhere else. It can only bind to this target site that you've indicated. Once that guide RNA identifies the site that you want mutated, then this enzyme, Cas9, makes a double strand cut at that point. So that is a mutation, and it's very site specific. Now, what you do with that cut is up to you. You can put something in it, leave something out. You can manipulate this cut point any way you wish. The important point is that you know what you're doing. You know what you've put in this site or what you've taken out. And therefore, if the mutation you've made is not carcinogenic, then CRISPR would not cause cancer. Is that clear? Any question about that? Someone asks, how are GMOs used? Well, one example I gave in class was golden rice, which rice does not have a pathway for synthesis of vitamin A, and vitamin A is required to prevent blindness in many countries of the world. So to make golden rice, the researchers took 2 genes from 2 different bacteria, put them into rice, so that now rice has a pathway for vitamin A production. And the reason it's golden, the reason it has a yellow color, is that's the color of vitamin A. And people who have vitamin A deficiency, if they eat golden rice, it remedies their deficiency. So that's one application of a GMO. GMOs are used commonly in agriculture to reduce the need for chemical pesticides like insecticide, herbicides, and fungicides. They can be used to make plants resistant to salty soils, resistant to cold or hot temperatures. Can be used to insert genes to increase yield. And this hasn't actually happened to my knowledge, but a proposed use for GMOs would be to put a vaccine, a protein, in a food, like an apple or a banana, so that when you eat it, you vaccinate yourself. Interesting idea. I haven't heard that it's ever been made to work. Okay, we're almost done. We're almost down to Mendelian. So the last question that I have here is on thermodynamics, the first and second laws. The first law is that energy is neither created or destroyed, it only changes form. The forms of energy are potential and kinetic. So potential energy is like a boulder sitting still on a hill. When it rolls down the hill, it now becomes kinetic energy because it can exert a force. The second law is of particular relevance in biology because all organized systems are subject to entropy. Entropy is a tendency toward disorder. So in order to maintain the organization of a system, it requires energy input. So living things, being organized systems, require a constant input of energy. And if they don't get it, they die. All the enzyme reactions stop if they don't have a constant input of energy. So that's the take-home message about thermodynamics. Okay, it is now 5 minutes to 2, and Scott's workshop starts at 2, so I'm going to sign off. Sorry I kept you over time. I will make a PDF of this document and send that out this afternoon.
Sarah, I have a question, please. Yeah, quick one. I'm asking about the vitamins. It's some vitamins and supplies, it says, like boost your energy, whatever. I have a question. why sometimes when you took like some supplements or vitamins, your body gonna be crash and you need to eat something, or you see your hand is shaking, whatever. How this work, I don't know.
Not sure I understand your question. You're saying that if you eat something, you start to shake?
No, if you took some vitamins or some supplies, you understand what I'm saying, like B12, whatever.
Yeah.
Why? I mean, after that, you're gonna be like a crash, and you need to eat food to. I don't know why.
Oh, I think that the answer to your question is you don't want a high concentration of a chemical in your stomach alone. In other words, if you take a pill of vitamin B12, it's a very high concentration of vitamin B12. When that dissolves in an empty stomach, could injure the lining of your stomach. So if you have a little food there, then you don't have such a high, the vitamin B12 gets diluted by the food, and the lining of your stomach is protected from this high concentration of chemical.
Okay, got it. Thank you, sir.
Okay, anything else? I'm going to stop sharing.